In chapter 9 now, we're going to go back to B cells and look at the second arm of the immune system, or the humoral immunity. We looked at cellular immunity or cell-mediated immunity with T cells in chapter 8, and now we'll look at B cells and how they play a role in the immune response. So let's go ahead and kind of break down what this is going to look like. It's a big set of slides. So I think there's over 50 slides. So I'm actually going to break this up into 4 different lectures. And the first 2 will look at one piece, which is on the left here, where it's going to examine the cellular interactions in that secondary lymphoid tissue, in those lymph nodes, in the gut, MALT, spleen, that lead to the mass production of antibody. And then we'll take a look at how that antibody is improved upon with each, through the development of plasma cells. And then we're going to take and break up the second part here on the right into 2 other additional lectures that looks at how antibody is used to control infection and then eventually terminate an infection. So it's everything to do with B cells and antibodies and how they are activated, made, and worked. That's really what chapter 9 is all about. So let's start out with looking at how antibody is produced by B lymphocytes, or rather by plasma cells, if we want to get right down into it. So we'll start with an anatomy of the lymph node. So we've seen this picture before. We looked at it when we were looking at T cells activation. But now, instead of focusing on the T cell area of the lymph node, we're going to move out just a little bit further and look at what's happening in the B cell area. So this is going to be packed with mature but naive B cells, and they are going to be interacting with follicular dendritic cells as well. So let's just take a peek here, and I gotta move this one thing that's kind of in my way. Oh, go away. Sorry. Oh, there's a little carrot I was looking for. Okay, so in topic 6, or in chapter 6, we looked at how B cells, when they're going through positive selection, they enter secondary lymphoid tissue and they have to get a survival signal from those follicular dendritic cells. And if they do not receive that survival signal from the follicular dendritic cell, they get arrested and they don't mature any further. And so that process of positive selection is a step towards B cell maturation. Now, those follicular dendritic cells, when they're interacting with B cells, they're actually going to be displaying antigen in an intact form. It's not processed, it's not presented like we've seen with antigen presenting cells, but rather they're going to be showing, or they will have grabbed some antigen and they will have it out for B cell receptors to interact with. Now, this will come in through the lymph, so that fluid that's flowing through the lymphatic vessels will have these antigens in it, because it's systemic, it's going throughout the body, and we'll bring in antigens. Follicular dendritic cells will pick it up and then we'll hold it, and then lots of times that antigen is going to have complement on it, because during the innate immune response, complement was activated either by way of the alternative pathway or lectin binding pathway. And those follicular dendritic cells will recognize the complement and grab that antigen. And then it will hold that antigen in the B cell area of the lymph node. So then we have B cells that are circulating through the lymph node, will then move into, first they'll start out in that T cell area. They'll move, because that's where they come in, usually through this high endothelial venule, and they'll come in and they'll move and migrate to the B cell area of the lymph node. They want to get to club B and they want to get that follicular dendritic cell, right? So then in that follicle, the naive, mature B cells are going to interact with those follicular dendritic cells that have antigen deposited on them. If the B cell receptor recognizes that specific antigen, then signaling to the B cell is going to be initiated. If not, then the B cell is like, well, didn't find my antigen, so then it will leave the lymph node. When a B cell finds its antigen, it's going to be through the B cell receptor, which is probably going to be an IgM molecule, because IgM is on the surface of a naive, mature B cell. And when more than one IgM molecule becomes activated, it's called cross-linking. So here is an image up here in the corner, left corner here, where we have our antigen, whether it's on the follicular dendritic cell or it's a bacteria cell, where the B cell is going to be engaging with antigen. We have multiple engagement points happening, and that's called cross-linking. When this cross-linking happens, then the signal is sent down through the cytoplasm of the B cell, which will turn on transcription factors so that there can be other proteins made that will signal the B cell to become activated. And so this clustering or this aggregation, this cross-linking of B cell receptors is needed in order for Ig alpha and Ig beta to get the signals to send down through the cytoplasm. And so this is the signaling cascade is started when that cross-linking happens. But just like what we saw with T cells, cross-linking is not just enough. It's definitely necessary, but there has to be a secondary signal that gets sent to fully activate this naive, mature B cell. And so the B cell actually has a co-receptor. And so it's made up of 3 distinct proteins. We have complement receptor 2, sometimes that's called CD21, CD19, and CD81. And collectively, these 3 proteins together function then as the B cell co-receptor. This receptor is actually going to bind to complement. So complement receptor is where the CR2 comes in, but it can be called CD21. It's the same, we can put or CD21. So these 3 proteins together will recognize complement along with the B cell receptors being cross-linked. Then that will allow for the signals to be sent through Ig alpha, Ig beta to increase the B cell's overall sensitivity to antigen and eventually become activated. So let's look at that in a diagram fashion. Antigen, though, can be attached to the surface of a pathogen, or it could potentially be soluble as well. So the first frame over here is showing when it's attached to a pathogen, where you have the bacteria cell that has multiple epitopes on its surface. And those epitopes are probably going to be repeated, and so they're going to be the same. And so you have antigen recognition of the same antigen specificity, but that's going to allow that cross-linking to occur of 2 different antigen binding sites. But then you also have the complement receptor portion recognizing complement on the surface of the protein. Here it's CD3, it could be C3b, C3d, C3b. Either way, it's deposited complement on the surface of the pathogen, and that is being recognized. And so we have our 1 signal here and our 2 signal here, and then that allows the transcription factors to be turned on to allow the B cell to become fully activated. Now, the same thing can happen with soluble antigen. The difference is that there's no bacteria cell that these antigens are attached to. They're soluble, but you still have to have multiple antigen receptors engaged for the cross-linking to occur. And then we still have complement on the surface of the antigen. You still have that opsin in there. And this C3 or this complement, the CR2 will recognize the complement protein. And so you still get the secondary binding. So you have the one binding here, and you have the two binding there, and then those signals together will allow for a change in gene expression to allow B cells to become fully activated. Okay. Once antigen is then recognized, that B cell is not going to leave the secondary lymphoid tissue. It's like, okay, so I found my antigen, and I found it, in conjunction with this follicular dendritic cell. And, I'm just going to hang out in this B cell area of the lymph node and internalize the antigen. I'll process the antigen, and then I'll present it on my own MHC molecules. And in this case, we're looking at an MHC class 2 molecule. So this diagram just kind of shows all of this happening. So you have we're kind of looking at two different things here. This is what we saw happen in chapter 8. And now this up here is what we're talking about in chapter 9. But both things have to happen because we know T and B cells have to work together for the innate immune response. But in chapter 8, we saw the naive T cell come in to the T cell area of the lymph node, interact with a dendritic cell that is presenting antigen. We have antigen recognition by the T cell receptor. We have the co-receptor, in this case, CD4, recognizing MHC class 2. And we have the co-stimulatory molecule of CD28 binding B7. All of that is allowing T cells to become activated. At the same time, we have B cells coming in. They're naive. They're going through positive selection. They'll interact with the follicular dendritic cell, which will have antigen attached to it. Cross-linking will happen, and we'll get the secondary signal down through the co-receptor, the B cell co-receptor, to then activate the B cell. So then at this point, again, here's our split where we have chapter 8 down here. We have chapter 9 up here. Our T cells have become activated down here in the T cell area. And they are starting to go through proliferation. They're starting to differentiate into effector T cells. Our CD4 T cells, T helper 1, T helper 2, T helper 17, they're probably all going to leave and go out to either site of infection, or they're going to go out and do their effector work. T follicular helper cells, though, are going to stay and migrate out to the B cell area. In chapter 9 here, we're talking about how B cells, then, we have activation happening because cross-linking is happening. We have our B cell co-receptors, so the 1-2 signals are being sent. B cells are starting to become activated. They'll internalize the antigen. They'll present it out on MHC class 2, waiting for a T cell to recognize it. Okay, so let's go back to an image we looked at actually this was probably in chapter 8 as well, last chapter. And let's walk through this step by step, and we'll put all the pieces together to hopefully create a good picture of what's happening at the cellular level. So during a primary immune response, we know that dendritic cells are the cell that is going to be in charge of activating T cells. So we have our bacteria gain access through a break in the skin, in this example. Dendritic cells live in the skin because they're sentinel cells waiting. They pick up pathogen. They end up migrating into the lymph node. Now we have dendritic cells that are in the lymph node, the draining lymph node, that are processing antigen that they picked up, and now they're presenting antigen to T cells. This is our dendritic cell, is presenting antigen in conjunction with MHC class 2 in this example to a T cell receptor and CD4 as the co-receptor. That's our first signal. We have CD8 CD28 receiving this co-stimulatory signal of B7 from the dendritic cell as our second signal. And then that is going to trigger the T cell to proliferate, make many more cells, and differentiate based on the types of cytokines that are driving the production of T cells. So then from this position, we have many T cells, right? Many T cells that will then go on to differentiate into, we're looking at T helper cells in this case, or CD4 positive T cells, into these four and potentially a Treg cell, but we're not worried about that right now. So these cells then differentiate based on the type of cytokine that is polarizing to either an inflammatory type response where macrophages are going to be activated. If it is a potential where more is a bacterial infection and more neutrophils are needed, then neutrophils will be enhanced if Th17 cells are proliferated or differentiated into. We're probably not going to have a very large presence of T helper 2 cells because it doesn't seem like there's a parasite. So likely these are going to be minimal to none in this case. But we do have some T follicular helper cells that are going to be created out of this differentiation. These 2 cells are going to leave and go out and interact with macrophages and with neutrophils and get them to be better at phagocytosis at the site of infection. But the T follicular helper cells are going to be retained in the lymph node because they are the cells that will then interact with the B cells to drive the humoral response of adaptive immunity. So let's follow these T follicular helper cells where they go into the B cell zone then of the adaptive immune or of the lymph node. Again, we're looking at a primary immune response or the first time that somebody is exposed to a certain pathogen. And the activation of almost all, just take that, there's always exceptions, but almost all naive B cells is absolutely dependent on a T follicular helper cell. A B cell without its T follicular helper cell isn't going to do much. And it is this interaction, multi-stepped, right? We have antigen being recognized in the conjunction of MHC class 2 molecules. We have CD4 recognizing, here's our T cell receptor. And then we have also CD40 ligand on the T cell is binding to the CD4 receptor or CD40 receptor on the B cell. So all of these signals then will allow the T follicular helper cell to start secreting cytokines, which we're probably looking at like IL-5 or maybe IL-21, maybe IL-4, depending on what type of infection we're looking at. But maybe IL-21 is probably the better one. But these are all going to send signals to the B cell to become activated, to proliferate, and then eventually to differentiate. Okay, now, because I said almost all B cells, there is a subset of B cells called B1 cells that actually do not require any T cell help. They do not undergo isotype switching, and they don't go through the process of affinity maturation. They will produce antibody, that's IgM, and it will recognize repetitive patterns on antigens, or sorry, on pathogens. So repetitive epitopes on pathogens. And so they're okay. It's not great, but it's something. And we call these B cells Thymus-independent antigens. Well, they call these antigens Thymus-independent antigens, but it's kind of a misnomer because it's actually the B cells don't require, they don't require the T cell help. So it's not that the antigen doesn't need the thymus, it's that the B cell doesn't need the T cells to recognize the antigen, or therefore the B cells don't need the thymus to develop a T cell to help the B cell. So it's kind of a crazy name, but just know that there is a set of B cells called B1 cells. We typically see them in developing fetuses and not so much in adults. But do you remember, if you're in my class on campus here, you probably can recall when we talked about DeGeorge syndrome in our case study, that even patients that have complete DeGeorge syndrome and do not make a thymus at all still have some low level of antibody. It was IgM, but it was there, and likely it's because it was produced from these B1 cells. Okay, so that is where, let's see here if I can get my mouse. There we go. That's where I want to stop then for this first lecture. We'll pick up and we'll look at continued what happens to B cells after they get T cell help to become activated.